Okay, hi everyone. We're starting a new chapter. This is on a chapter on taxonomy, chapter 10. Taxonomy is the science of the naming and classification of organisms. And in this painting, you can see Linnaeus, who was the father of taxonomy and actually named quite a number of organisms himself. He developed binomial nomenclature, the two-part naming system, so binomial, so two names. So we currently name things calling them by their genus and species name. So like for example, Escherichia coli. Escherichia is the genus, coli is the species. We always capitalize the genus and the species is always lowercase. And here in print on this slide, you can see that it is italicized. If you were writing up a little homework assignment and you were printing it, you would underline it. Okay, but in print like this, if you were typing it up, you would highlight it and then italicize it. So the classification of organisms is based on their evolutionary history or their phylogeny. And this makes this field really a dynamic field because we'll talk about what constitutes a species, but with bacteria, species were based on what they looked like, and they were grouped together based on their morphological characteristics. But once the DNA technology for sequencing took off, and we started to sequence the genomes of bacteria, that really gave us a lot of insight into the evolutionary history of these organisms, because we could actually look at their DNA sequence and look for similarities and group them together based on that. So while, in some textbooks over the years, the information hasn't changed a whole lot, like 20 years ago, the textbooks that were written, the taxonomy is completely different at this point. Okay, so it's really a changing and dynamic field. So I mentioned, what constitutes a species, and for eukaryotic species, okay, like for instance for humans, the species is classified as a group of organisms that can interbreed. And so even though when we look at humans, they can look quite different, right? Different hair color, etc. They can interbreed, and so this is how we would classify a eukaryotic species. So this is what's referred to as a biological species concept, and that is not something that you can use for prokaryotes, for bacteria, because they divide by binary fission, which is an asexual process. So for bacteria, we would look more at what's referred to as a morphological species concept, what they look like, okay, what their characteristics are, and that would be how we would define a species. Okay, so a prokaryotic species is a population of cells with similar characteristics that would be derived from a single bacterium. So that would be a clonal population. This is the species concept, the morphological species concept that would be used to classify, say, for instance, fossils. Okay, so we don't really know how a fossil would possibly be able to interbreed, but we can look at the morphology, right? And classify it in that way by its similar characteristics. So I mentioned DNA technology, DNA sequencing technology. So when we started to sequence the DNA of bacteria, that really gave us huge insight into the evolutionary history of those organisms, because we could actually see similar sequences between species and figure out how related or unrelated they were. So we may have thought that some species were highly related, and then it turned out that they were not. Okay, when we looked at the actual DNA sequence. This is the classification hierarchy for starting with the kingdom. Okay, so the way that life is currently organized is into the 3 domains. So the domains would actually overarch the kingdom. It would start with domain, and then it would go to kingdom. So here we have the classification hierarchy going from kingdom to phylum, class, order, family, genus, and species. And then below the species is, if you look at the hierarchy to the right, is the strain. Okay, so within a species, you can have several strains. And those are usually indicated by numbers or letters after the species name. Just looking at this hierarchy right here, you can see that the order, Spirochaetales, it has the suffix ales, and then the family, Spirochaetaceae, okay, has the suffix acea, okay, ACA, and that is similar for the family Enterobacteriaceae, which we will actually be studying in the lab. So within that family, the family Enterobacteriaceae, there are a lot of genera, and each of those genera have numerous species. Okay, some more than others, right? Some genera might only have 1 or 2 species. Others might have thousands, like in the case of Salmonella from the family Enterobacteriaceae. Okay, Salmonella would be the genus. There are actually 2,000 different species of Salmonella. Okay, and then if there were numbers or letters beyond that species, that would be considered the strain. So in the same group, okay, as you move down the hierarchy toward the species, those organisms would be becoming more highly related. So like, for instance, the genera in the family Enterobacteriaceae are more highly related than they would be to genera from a different family. Below the species is the strain. Okay, so like, for example, E. coli O157H7, okay, those numbers and letters below the coli, the species, indicate the strain. Okay, and this is like the so-called bad E. coli, right? That causes the hemorrhagic diarrhea and can be associated with contaminated food. Okay, so but there are different strains of E. coli which are not pathogenic. Okay, this is a true pathogen that causes disease, but other strains of E. coli live in our gut, and we actually have a very positive, mutualistic, symbiotic relationship with them where we benefit them and they benefit us, okay, by producing vitamin K. So the orders I mentioned end in ALES, okay, and the family ends in the suffix ACA. And then I gave you that little mnemonic device, okay, and other people have other mnemonic devices for this, but how can you remember the hierarchy? Keep pots clean or families get sick. So kingdom, phylum, class, order, family, genus, species. All right. So before the three domains, okay, and we'll look at that in just a minute, life was organized into these 5 kingdoms, okay, Plantae, fungi, Animalia, Protista, and Monera. Plantae, fungi, and Animalia are still recognized, and Protista is currently being divided into a bunch of kingdoms, okay, or genetically related clades, and they are all part of the domain Eukarya. Okay, so they're all eukaryotic organisms. Monera, on the other hand, which contained all of the prokaryotes, has actually been divided into 2 domains. Okay, so the domain bacteria and also the domain archaea. Okay, and so we'll look at all of that in just a minute and talk about all of those different organisms. Okay, so I mentioned that the kingdom Monera is no longer recognized. It contained all of the prokaryotic organisms, including the eubacteria, okay, the true bacteria that are now part of the domain bacteria, and the archaeans, okay, which we'll talk about in just a minute when we talk about the domain archaea. But this one kingdom now is 2 of the 3 domains. So it was just 1 out of 5 of the kingdoms, but now it's 2 of the 3 domains. And that actually more accurately reflects the distribution of species on Earth. So these species are more numerous than the eukaryotic species, even though that we can't see them, right? Okay, but we can see the eukaryotic species, but they are more numerous, and so by organizing these species into 2 of the 3 domains, we are actually more accurately reflecting this. Okay, so you can probably see some bacteria here that you recognize, like the gram-positive and the gram-negative bacteria, the spirochetes. Mycoplasma are wall-less bacteria that are pretty interesting. The cyanobacteria, okay, are formerly referred to as the blue-green algae. And then the archaeobacteria, okay, would be the archaeans, which we'll talk more about. The kingdom Protista I mentioned is no longer recognized. When we look at the evolution of taxonomy, we'll see that in the beginning it started out as the plants and the animals, and then there were some organisms that didn't fit into either the plants or the animals, and so they developed a third kingdom, which was the kingdom Protista, and anything that wasn't a plant or an animal, okay, that were like the so-called unicellular plants and animals, like for instance, for an example of a unicellular animal like an amoeba, or a unicellular plant would be like algae, got put into this kind of catch-all kingdom, which was the kingdom Protista. But this is actually really a nutritionally diverse kingdom. Okay, like I just mentioned with the amoeba and the algae, they are, one is performing photosynthesis, right? And then the other one would be a heterotroph, a chemoheterotroph, so obtaining its nutrition by engulfing other organisms, so getting that nutrition ready made, whereas the photosynthetic organism is making its nutrition, it's making its carbohydrates. So these organisms that are so nutritionally diverse are actually being divided into clades, okay, which would be genetically related groups, which could develop into many more kingdoms. So the kingdom fungi is still recognized, and we've mentioned the kingdom fungi before, so it contains all of the multicellular molds, and the unicellular yeasts, and the macroscopic mushrooms, okay? And even though the mushrooms kind of look like plants and they have, kind of plant anatomical parts, right? They are not plants. They do not perform photosynthesis. They are all decomposers. So they're all chemoheterotrophs. Okay, so they get their nutrition ready-made, versus a plant, which would be a photoautotroph. Okay, so it would use the light, okay, photo, as its energy source, and auto, okay, it would make its own carbohydrate containing, it would make its own carbohydrates. The kingdom plantae are all photoautotrophs, okay, so they perform photosynthesis, they fix carbon dioxide from the atmosphere, they play a very important role in a self-sustaining ecosystem, right? They are the producers, and they contain all of the plants, from the most primitive mosses to the most highly evolved flowering plants and conifers. Kingdom Animalia, right, are all multicellular. Okay, they're all chemoheterotrophs. Okay, they all obtain nutrients via some type of mouth. Okay, maybe very primitive. In some of the more primitive phyla, but they all obtain their nutrients via some type of mouth and are all chemoheterotrophic. So life is currently classified into the three domains. The WOSI method of classification. So there are two prokaryotic domains, okay, which would be domain bacteria and domain archaea. And then there is one eukaryotic domain, okay, domain eukarya. And we can see that domain eukarya contains 4 of the 5 kingdoms. Okay, so kingdom fungi, plantae, animalia, and the kingdom Monera is no longer recognized, has been divided into the 2 domains, bacteria and archaea. It's really interesting to look at the evolution here of the domains and the kingdoms. So with Linnaeus, it started out with just the plants and the animals. And then you can see, okay, that some of the animals and some of the plants got put into a third kingdom, which was the kingdom Protista, right? So these would be the unicellular plants and animals, so to speak. Okay, so the unicellular eukaryotes, okay, like the amoeba or like, you know, say for instance a paramecium, okay. So that made up the third kingdom. And then you can see from there, okay, that some of those, okay, so some of those organisms from Protista were actually fungi, okay, could have been like the yeast, for example, the unicellular yeast. Some of them remained in the kingdom Protista, okay, and those are very nutritionally diverse organisms. And then some of them turned out to be bacteria, okay, and so they got put into the kingdom Monera. From the plants, okay, the multicellular, perhaps, mushrooms were actually reclassified into the kingdom fungi. All right, so you can see the 5 kingdoms there. All right, and some of them now are no longer recognized, and life is organized into the 3 domains, okay, bacteria, archaea, and eukarya, okay, and 2 of those 3 domains are actually prokaryotic, okay, these simpler organisms. So the domain bacteria, okay, would be our first prokaryotic kingdom, and we talked about peptidoglycan, okay, and I think at the time in chapter 4 I mentioned to you that peptidoglycan was referred to as the murine layer. So the organisms in domain bacteria contain peptidoglycan in their cell wall. All right, that is one of the major differences between domain bacteria organisms and domain archaea organisms. So domain archaea organisms do not contain peptidoglycan. They are prokaryotes, but they do not contain peptidoglycan in their cell wall. And so they have what's referred to as the pseudomurine layer. So domain bacteria is going to contain all of the prokaryotic pathogens of humans. Okay, that's not to say that all bacteria are pathogenic, all right, because they're not. But as opposed to the domain archaea, okay, domain bacteria would contain the prokaryotic pathogens of humans. Domain archaea actually contains a lot of the so-called extremophile organisms. I think I mentioned those before. So these would be organisms that would live in places that you think, you know, would exclude life, right? That nothing would live there. like for example, I think I mentioned the Dead Sea, right? Okay, the Dead Sea that they thought was completely dead. There are organisms, there are halophilic organisms that can live in the Dead Sea. Okay, they can live in that 30% salt concentration. This is a black smoker vent, which is deep in the ocean. It's like a volcanic vent that spews out this kind of sulfur gas, and so this is deep in the ocean below where the sunlight penetrates. Okay, so you wouldn't have like any algae growing way down there because they require the sun, right? They're photosynthetic, so they require the sun as their energy source. So this is like below the photic zone where it's dark. And these organisms can actually oxidize the sulfur for energy. So they can use the sulfur as their energy source and grow and divide, and they can actually be a source of food for other organisms. So they can play a very important role in the food web deep in the ocean. Okay, so this would be an example of an archaean that would be like an extremophile organism that would be living in a place where you think, nothing would live. So domain archaea, the organisms do not contain peptidoglycan. They're prokaryotic, but they do not contain peptidoglycan in their cell walls, so they have what is referred to as a pseudomurine layer. Here are some other examples of extremophile organisms. Okay, I mentioned the extreme halophiles. They have actually come to require a high salt concentration for survival. So there's a lot of organisms like, for instance, E. coli that we mentioned that live in our gut that would not be able to live at like a 3% salt concentration. But these extreme halophiles that can live, like for example, in the Dead Sea, okay, which has a greater than 15% salt concentration, they have actually come to require that high salt concentration. The hyperthermophiles, okay, so a thermophilic organism is an organism that can grow in a hot environment, like for instance, the organisms that can grow in a compost heap, okay, which is like 60 degrees Celsius, okay, and sometimes those smoke, right, and they can even catch on fire. But the hyperthermophiles would be organisms that could grow almost in like boiling water. Okay, they can grow almost in like a boiling hot springs. So they can grow in extremely hot temperatures, like maybe 90 degrees Celsius. An example here is the organism Thermus aquaticus, which actually has turned out to be a really valuable resource for biotechnology. Some of you may recognize that name. Thermus aquaticus is the organism that Taq polymerase is isolated from, and we'll talk more about that when we get to our biotechnology chapter. But these are really amazing organisms because when you think about, what that temperature would do to our proteins, okay, it would denature them, right? It would denature our proteins. It would break the hydrogen bonds, and our proteins would, unravel and, bond with other unraveled proteins and fall out of solution. They would become denatured. These organisms can actually grow and divide and require they probably won't grow at room temperature or at body temperature. They require this extremely hot environment for their development. Another example of an extremophile would be the methanogens, which can produce methane from carbon dioxide and hydrogen. Okay, so these are extremophile organisms, very ancient organisms that are part of the domain Archaea. Here's an example of a question. Okay, I will let you think about that and refer to your notes and to come up with the answer. Here's another example of a question. Okay, they might seem simple now that we just reviewed the material, but remember when you're taking an exam, okay, this is going to pop up out of nowhere after you're, maybe answering a question on microscopy. Okay, so that would be an example of another question. I just want to talk about the steps of a complete taxonomic study. So basically, you would need to establish a pure culture and characterize the pure culture for a complete taxonomic study. Okay, so we talked a little bit about how Koch established his pure cultures, right, by streaking, okay, and how he developed the streak plate method. So if you were to acquire a culture from the environment, it would probably be a mixed culture, and you would need to streak to isolate your pure culture. Okay, so a pure culture is going to be a well-isolated colony, okay, which is not always a pure culture because it could be sitting on top of another colony, okay, that maybe we couldn't see. But after streaking maybe multiple times, okay, our primary streak plate and a secondary streak plate, and we're pretty confident that we have a well-isolated colony where all of the bacteria in that colony are derived from a single bacterium. That would be our pure culture, and then we could go on to characterize that pure culture microscopically, culturally, biochemically. We could look at specific chemical characteristics, and then we could also look at genetic characteristics, including the sequence, okay, which may give us a lot of insight into what the organism actually is, or if it's an unidentified organism, what it's related to. So microscopically, in a taxonomic study, the best stain to do, okay, would not necessarily be a simple stain, because a simple stain is only giving you morphology and arrangement. But if you did a differential stain, like for example the Gram stain, you could see morphology and arrangement, as well as get more information about the cell wall composition, right? And by knowing morphology, arrangement, and gram type, okay, that would really narrow down your organism into a single taxonomic group. From there, okay, you might want to do, depending on, what you saw, okay, like say for instance, if you saw a gram-positive rod, okay, you might want to do an endospore stain, or you might want to check to see if it's acid fast, okay? And then there are also structural stains that we could do. Okay, we could look for a capsule or a flagellum. Okay, I think I described to you guys the problems with the flagellar stain. So we would probably determine motility by using a biochemical test, and we'll talk more about that toward the end of the chapter. End of the semester. Okay, so also, all right, it would be good to look at the cultural characteristics. Okay, so you've grown this colony on a petri dish. You could look at the colony characteristics. So you could look at the opacity of the colony. Is it opaque? Is it translucent? You could see lines, light through it. Or is it transparent? Okay, you can completely see through it. What are the margins of the colony? Okay, or the edge of the colony? Is it a smooth edge? Okay, which would be considered an entire edge, or is the edge undulating? Okay, or does it look lobate? All right, so these would all be words to describe the margins of the colony. Okay, how does the colony stack up on the surface of the plate? Okay, sometimes that's hard to see because they're so small, right? But if you looked straight down over the top of your petri dish, you can get an idea of how they're stacking up on the plate by looking at the density of the colony. So an umbonate colony is a colony that has a raised center. And if you were looking over the top of your petri dish, the center of the colony would look a little bit more dense, okay? And that would give you the indication that that was an umbonate colony, okay? Or it could be convex, all right, or etc. Okay, it could be different. You could also look at environmental characteristics. So, like for instance, what temperature your organism grows at. Okay, so it might prefer room temperature over body temperature, 37 degrees. It might not grow at body temperature. It might only grow at room temperature, or vice versa. Oxygen requirements. Is it an aerobe? Okay, does it like an oxygenated environment? Okay, or perhaps an oxygenated environment is toxic to it and it can only grow in an anaerobic environment in the absence of oxygen. You could look at pH requirements. Does it divide more frequently in acidic growth media, or does it divide more frequently in growth media that has a neutral pH? Okay, so you could look at environmental characteristics like that. Nutritional factors, okay, what's the energy source? Is it a phototroph? Is it a chemotroph? For us in the lab, okay, we don't really give you phototrophs for the unknowns. We would give you a chemotroph. This is a very important tool of taxonomy, and you guys will be making your own dichotomous keys later in the semester. But say, for instance, that we were looking at possibly trying to differentiate several genera from the same family. And for some families, like in these particular genera that we're looking at here, Shigella, Salmonella, Escherichia, Citrobacter, and Enterobacter, they are all from the family Enterobacteriaceae. And they all have very similar microscopic and cultural characteristics. So, like for instance, they would all be gram-negative bacilli, okay? They might be diplobacilli, okay, or they might just occur singly under the microscope. And they would all be pink after the gram stain. As far as their cultural characteristics are concerned, they would all be, as far as their colony characteristics are concerned, they would all look translucent, okay, on a petri dish. They, as far as their cultural characteristics are concerned, they would all be facultative anaerobes, okay, which means that they can live in an aerobic environment or an anaerobic environment. They would all prefer body temperature, okay, since they live in the gut of humans or animals. And so those are all very similar. So we would need to go to the next step in our taxonomic study, which would be the biochemical characteristics, and look at how they metabolize different biochemical substrates. Okay, so in the first step here, we're asking the question, can they ferment lactose? So we would inoculate them into a lactose broth, which has a pH indicator. And if they can ferment the lactose, the pH indicator would turn a color, okay? So here, the pH indicator would be phenol red. And at a neutral pH, the media would be red. But if they fermented lactose, the media would turn yellow. So Escherichia, Citrobacter, and Enterobacter all ferment lactose. Okay, and that's usually true of the non-pathogens, that they ferment lactose, and then the lactose non-fermenters are the pathogens. Okay, so like for instance, Shigella and Salmonella do not ferment lactose. Okay, so we could, we have this unknown bacterium. We could inoculate a lactose broth, come back the next day, all right, say it's not fermented. Okay, then we suspect, okay, we have Shigella or we have Salmonella. Okay, so now we need to find another test when we're creating this dichotomous key that will differentiate Shigella and Salmonella. Okay, we don't want to use a test where they both have the same result, because then we won't be able to know what our unknown is. But if we find a test that, like, Shigella is negative for, Salmonella is positive for, okay, then, all right, when we test our unknown and we get our result, we will know whether we have Shigella or Salmonella. So it turns out that by using this test, Simmons citrate agar, okay, to determine whether a bacterium can use citric acid as its sole carbon source, right, we can differentiate Shigella and Salmonella. Shigella, and again, this would be another biochemical test that has a pH indicator in it. So we would inoculate our bacteria and then look for a color change, okay, which would indicate a positive result. No color change would indicate a negative result. And then we would be able to determine, okay, what our unknown bacterium is. Okay, so this is an example of a dichotomous key, which is a very important tool of taxonomy. These are really neat rapid identification tests where, okay, we were just talking about lactose fermentation. And if you look at the numbers, right, it's number 6. Okay, so number 6, you can see, okay, on the uninoculated control that's on the very, very bottom, the 6th little compartment right there is red. Okay, and if it turns yellow, like it did for the E. coli, okay, in the test above that, the uninoculated control, the test above that is E. coli. If it turns yellow, that means that it fermented lactose. Okay, and then you can see for Proteus vulgaris, okay, it remained red, right? Number 6 remained red. Okay, so Proteus vulgaris is a lactose non-fermenter. The very, very end test, number 12, okay, that is citrate, all right? If the organism can use citrate as its sole carbon source, okay, the pH indicator turns the media blue, all right? So you can see for Enterobacter aerogenes, it is positive for citrate. Okay, so these are really, they're called the enterotube, and you can see on the end that you can screw on and off those caps. And basically there's like an inoculating loop in there. And so you would screw off the cap on one end and use it like a loop to pick up a bacterial colony. And then you would pull that loop all the way through all of those compartments, all of those tests. And then you would push it back through, okay, so that you're inoculating every single compartment with that bacteria. And then you would screw the lid back on and you would incubate that test in the incubator, okay, come back the next day or 48 hours later and read it, okay, and you can read by the color changes which of the tests are positive and which are negative. There are some, it says use up to 15 biochemical tests. So there are some tests, okay, like for instance, number 4, okay, you would read it for sulfur, and then you could also open that test up and add some drops of a reagent to look for indole production. Okay, so that particular little box, number 4, is actually 2 tests. Okay, so up to 15 biochemical tests in this one enterotube, which would be a rapid identification test. Methods is misspelled. Okay, so another step on our taxonomic study would be chemical characteristics, and you could do a chemical analysis of, like, say for instance, the whole cell fatty acids, and fatty acids are not constant for a particular species. So you could create some type of fatty acid profile and compare it to a known organism. And you could do, some type of chemical analysis like that. I gave us, for our example, a more biological type of example where we're looking at, kind of biochemical characteristics in this ELISA test. So an ELISA test stands for enzyme-linked immunosorbent assay. And in this particular assay, we have known antibodies adhered to the well of a microplate. And so those antibodies would be raised against a particular organism. And when we add an unknown bacterium, okay, if it actually binds to that antibody, okay, then we would have identification of that organism, all right? Because if it binds to the antibody is specific for a particular bacterium. So if it binds and then in this direct ELISA, okay, where we're sandwiching the antigen between two antibodies, okay, we would have a second antibody, okay, raised against a second antigenic site or epitope, okay, if that means anything to you right now. Right, we'll talk more about that when we get to chapters 16 and 17. But we would have a second antibody that would bind to a second site on that particular bacterium that would be bound to an enzyme. Okay, so that little star is the enzyme. And when we add a colorless substrate, if that second antibody is present, okay, and it's only going to be present if the antigen is present, the enzyme will turn the colorless substrate into a colored substrate, and we will get a positive test. Okay, now imagine it's negative. Okay, so that was the scenario for the positive. Okay, for the negative, the antigen does not bind. Okay, which means that the second antibody with the enzyme does not bind. You add the colorless substrate, it doesn't turn color because the enzyme isn't present. Okay, and that would be a negative well. All right, but we can actually get identification by doing an ELISA test. For genetic characteristics, okay, and we could just sequence the DNA, but ribosomal gene sequencing also gives you some really important information without having to sequence the entire genome, okay, because the entire genome can be quite large, whereas the RNA, the rRNA, the ribosomal RNA that's part of the ribosome is just a single or several genes. Okay, so we could do ribosomal gene sequencing. The reason why the ribosomal gene, okay, for the rRNA, the ribosomal RNA, is used is because over the course of evolutionary history, there have been very few changes to these ribosomal RNA genes. And the reason why is because the rRNA in the ribosome binds to the mRNA, okay, to position it in the ribosome for translation into the protein. And if there is a change in that sequence, okay, it could be lethal to the organism because if you have a change, okay, a mutation in the rRNA and it affects the binding of the mRNA and that mRNA is not translated into protein, that could kill the bacterium. Okay, so that's negative selection, right? Okay, so you're selecting against that particular mutation. And so over the course of evolution, there have been very, very few changes to these ribosomal RNA sequences. And so you can actually, by looking at the sequence here, determine a genetic relationship. Okay, there are signature sequences within genera. Okay, so you can really get some important information there. I also wanted to talk about DNA base composition. So you might read or you may have read in the book about percent G plus C. This is what we would call a fixed property, all right? So if you have a DNA sequence, okay, and these are clonal populations that we're talking about, guanine is going to base pair with cytosine, okay, adenine base pairs with thymine, all right? And you're going to have a certain number of G's, okay, base paired with C's, right? A certain number of A's base paired with T's. And that is not going to change. Okay, so if we look at the percent G plus C ratio, and two organisms are closely related, they're going to have similar G plus C percentages. If they have different G plus C ratios, okay, more than 10% difference, then you can definitely conclude that they are unrelated. All right. So like if one organism has 40% G plus C, the other one has 60% G plus C, okay, those organisms are not related. But, okay, just a word of caution here. Some organisms can have very similar G plus C percentages and not be related at all. Okay, and the example that I give there is humans and rice have the identical G plus C percentages, and they are not related, of course. Okay.